Sponsored by RxCardClub.com. Get a free prescription drug card and save up to 85% on prescription drugs. RxCardClub.com. The 5 types of antibodies. Different types of antibodies are produced by your immune system. These 5 antibodies help fight disease by detecting viruses, bacteria, and other pathogens, disease-causing microorganisms, and working to destroy them. Each has distinct methods of defending your body against disease and infection. /R/N/R/N harmful infectious organisms are identified as invaders due to their antigens, distinct molecules on their surface. Each antibody produced by your immune system binds to a specific antigen and then either destroys the pathogen or tags it so other immune cells will recognize it. /R/N/R/N antibodies are also known as immunoglobulins, IgG. Immuno describes immunity and globulin describes protein. They are produced by B cells, a specific type of white blood cell, WBC, that originates in the bone marrow. /R/N/R/N While there are only 5 main types of antibodies, each can have a different binding site that matches a specific antigen. Your body can produce an infinite number of binding sites to bind to antigens. /R/N/R/N immunoglobulin G, IgG, accounts for around 75% of all antibodies in the human body. Depending on the antigen, IgG can either tag a pathogen so other immune cells and proteins will recognize it, or it can activate the complement system to directly destroy the microorganism. /R/N/R/N can sometimes trigger an undesirable response in people with autoimmune diseases, in which the immune system inadvertently attacks its own cells and tissues. /R/N/R/N immunoglobulin A, IgA, is primarily found in mucosal tissues, such as those in the mouth, vagina, and intestines, as well as in saliva, tears, and breast milk. It accounts for 15% of all antibodies in the human body and is produced by B cells and secreted from the lamina propria, a thin layer within mucosal tissues. /R/N/R/N IgA is one of the body's first-line defenses against infection. It binds to pathogens to tag them for destruction and prevents them from sticking to the epithelium, which lines the body's tissues. /R/N/R/N IgA is also associated with hypersensitive reactions in people with celiac disease and several other autoimmune disorders. /R/N/R/N An autoantibody is an antibody directed against the body's cells. Antibodies are designed to attack invading pathogens, like viruses and bacteria. Autoantibodies occur when the immune system becomes confused and mounts an assault on the body's own proteins. /R/N/R/N autoantibodies are involved in autoimmune diseases like rheumatoid arthritis, type 1 diabetes, multiple sclerosis, and lupus. /R/N/R/N immunoglobulin M, IM, is also one of the first antibodies recruited by the immune system to fight infection. IgM populations rise very quickly when the body is first confronted with an infectious organism, and then they plummet as IgG antibodies take over. IgM is also produced by B cells and, when bound to a pathogen, will spur other antibodies and immune cells into action. /R/N/R/N in addition to activating the immune response, a subset of IgM helps B cells remember a pathogen after it has been destroyed. If you were to become re-exposed to the pathogen later, your immune system should respond more quickly due to your memory B cells. /R/N/R/N immunoglobulin E, IgA, is the antibody responsible for the allergic response that is mostly found in the lungs, skin, and mucosal membranes. IgA is produced by B cells secreted by lymph nodes or other lymphoid tissues situated near the site of the allergen, a harmless substance that induces an allergic response. /R/N/R/N when IgA binds to an allergen, it triggers a cascade of events. Basophils and mast cells, which are subtypes of WBCs, degranulate, break open, and release histamine, an inflammatory compound, into the bloodstream. It is histamine that is responsible for many of the most common symptoms of allergy. /r/n/r/n IgA also helps to protect the body from parasitic infections, including helminths, parasitic worms. /r/n/r/n immunoglobulin D, IgD, is important in the early stages of the immune response. Unlike other antibodies, it does not actively circulate but instead binds to B cells to instigate the immune response. As a signaling antibody, IgD helps incite the release of frontline IgM to fight disease and infection. /r/n/r/n IgD accounts for only around 0.25% of antibodies in the human body. Despite its vital role in kickstarting the immune response, IgD is arguably the least understood antibody, with little known about how it might participate in other parts of the immune system. /r/n/r/n A monoclonal antibody, mAb, is a type of immune protein produced in a lab that binds to a specific protein on a cell, an antigen. As with naturally occurring antibodies, monoclonal antibodies stimulate the immune system to act against disease-causing agents. /r/n/r/n monoclonal antibodies are used to treat numerous health conditions, and many have multiple indications and can be used to treat different diseases. /r/n/r/n at least 25 monoclonal antibodies are licensed for different cancer treatments, including /r/n/r/n at least 20 different monoclonal antibodies treat autoimmune diseases like /r/n/r/n there are newer monoclonal antibodies that specifically target and treat diseases unrelated to cancer or autoimmunity. /r/n/r/n these include monoclonal antibodies licensed for the treatment of /r/n/r/n monoclonal antibodies, mAbs, are made using identical immune cells all cloned from a specific parent cell, while polyclonal antibodies, PAbs, are made from multiple immune cells. In addition to their origin, they fundamentally differ in specificity and utilization. /r/n/r/n because immunoglobulins are matched to a specific pathogen, they can be used to diagnose some diseases based on their unique structure. Antibody tests are used to detect disease-specific antibodies in a blood sample. /r/n/r/n antibody tests are available to diagnose, or help diagnose, a wide variety of infectious and autoimmune diseases, including /r/n/r/n antibody tests do not detect the actual pathogens that cause an infection, they detect the antibodies that are produced in response to the infection. A positive result means yes, the test has detected the antibody or antigen. A negative result means no, while borderline results are considered inconclusive. /r/n/r/n depending on the disease, it may take time for enough antibodies to be produced to reach detectable levels. If it's done too soon, during the early window period, the test may deliver a false negative result. /r/n/r/n some disorders are associated with a deficiency, but not from a specific IgG. For example, primary immunodeficiency disease, PID, is diagnosed with a deficiency of total IgG. /r/n/r/n an antibody test can confirm that an infection has taken place, as with COVID-19 or HIV, although it cannot tell you when. /r/n/r/n sometimes, immunoglobulin levels can be used to characterize the stage of an infection. Because IgM levels usually increase before the IgG response kicks in, a disease-specific IgM and IgG test can help determine whether an infection has occurred recently. For example, herpes simplex is an infection for which IgM and IgG tests can help determine the timing of the infection. /r/n/r/n in people with allergies, IgA tests can be used to confirm that an allergic response has occurred. /r/n/r/n the body has 5 different types of antibodies, also known as immunoglobulins. IgA, IgD, IgG, IgA, and IgM are different immunoglobulin isotypes. /r/n/r/n immunoglobulin A, IgA, defends against infection by binding to pathogens to tag them for destruction. IgD binds to B cells to kickstart the immune response. Ig binds to a pathogen or activates antibodies to destroy it. IgA fights parasitic infections and binds to allergens. IgM prompts the release of other antibodies to destroy pathogens and provides immunity to illnesses you have had or been vaccinated against. /R/N/R/N antibodies are produced in response to a disease or infection. They are not the disease or infection. Some antigen tests can detect an actual pathogen by its signature antigen. Some diseases can be diagnosed with either an antibody or antigen test. /R/N/R/N, rxcardclub.com. Get a free prescription drug card and save up to 80% on prescription drugs.